Mm.
Hearty welcome to you, sister.
Sister, please join in the link, sister, just to give the opening remarks for the today's webinar. We request our correspondent sister to join in the given link to give the opening remarks of today's webinar.
Yes, thank you, madam.
Thank you, sister.
Are you able to see me? I don't know exactly. Okay, anyway. Are you able to see me, madam? Hello? Are you able to hear me?
Yes, sister.
Yeah. Okay.
Yes,
So, very
sister.
good morning to everyone, and a hearty welcome to all my dear loving Nirmalites, students from various institutions, respected principal sir, Dr. Pamla Reddy, organizing secretary, Dr. Supriya madam, and other members of the Department of Pharmaceutics, as well as members of the Research, Innovation, and Ideation Cell. Today, we are very honored to have with us our esteemed resource person, Dr. Fani Kumar, a well-known person to us. It is a pleasure to see you here, sir, Dr. Fani Kumar, sir, after a long time. Nice to see you in this webinar. So, a hearty welcome to you, sir.
Thank you, madam.
First and foremost, I extend my sincere appreciation to organizing secretary, Mr. Supriya madam, and coordinators, Mr. Naresh Babu sir and Mrs. Sasikala madam. So now turning to today's webinar, the topic is neuroscience, cellular and molecular mechanisms of neuropeptides. So as we all know that neuropeptides are small protein-like molecules used by neurons to communicate with each other. They play a very crucial role in various physiological processes, including like pain modulation, stress response, or mood regulation and appetite control, etc. So understanding these cellular and molecular mechanisms of neuropeptides is essential for you students, especially who are pursuing science, pharmacology, and medicine. So today our distinguished guest, Dr. Panikumar, is with us. He is going to enlighten you, dear sisters, on how this knowledge can be beneficial in terms of research or drug development, clinical practice, and education pursuit. So I request you, dear students, to utilize this great opportunity. And once again, I thank everyone for your presence at this webinar. I wish you all the best and hope you make this day fruitful. Thank you and God bless you.
Thank you so much, Sister Yogita. Now, I would like to invite our principal to address the gathering.
Morning, Lord Secretary and Correspondent, Sister G. Kamala my colleagues, C.S. Supriya Madam, Naresh Babu Sir, Shikala Madam. And today's speaker, Dr. Panikumar Sir, so postdoctoral fellow from USA, and my dear students, very good morning to one and all. So today we have the diploma of pharmacy and pharmacy and diploma on pharmacy. There are many subjects we study. Okay, so majority of the subjects which are related to the pharmacology and therapy we study in the curriculum of pharmacy. But at the same time, we need to know about the outside the box, what is happening other than the curriculum. That will be helpful for us for our career growth. Because what are the things which you are going to study, that will be helpful at the time of R&D level. But if you want to go into the some other areas, especially in the kinetic departments, especially for the pharmacy graduates, we should know the thing what is happening other than out of box, other than the curriculum. So that's why we used to conduct the many seminars and workshops to the budding pharmacists to grab the knowledge other than the academics. That's why I request all the students to utilize this opportunity. Because after the graduation, there are so many opportunities you get. But during the academic itself, if you come to know about what is happening in the, especially in the kinetics departments, then you can come up in the life. Because now today's topic is neuroscience, neurotechnology. Because if you take the USA, especially USA, the majority of the people they are suffering from the neurological and psychiatric problems. Slowly it has come into the India also. Because of what are the mobiles or what are the things nowadays we are using. Because of that, or it may be due to the food problems also. Majorly, the people they are facing the neurological and psychological problem, because there are so many articles recently which was published, especially the usage of mobile phones in the India. Gradually, day by day, it is increasing. Because the 9th class students and 10th class students itself, they are using the highly sophisticated mobile phones. Because the parents are encouraging. So finally, what will happen leads to the psychiatric problems. Okay, so that's why. So in this regard, so I appreciate the team members of C.S. Supriya Naresh Babu sir and Shikala madam, other faculty members over here, for organizing such a type of webinars to the budding pharmacist. Because so especially this type of topics are very useful to the students those who are seriously looking into the abroad. And the students those who are seriously planning their future into the basic R&Ds. Such type of things are very important. That's why whenever you conduct any seminars and workshop, you have to utilize it. At least you should collect some of the important points. Okay. So in this regard, I welcome Dr. Fani sir. So after long time, we are seeing Dr. Fani sir. Welcome, sir. Welcome.
Thank you, sir.
Sir. So I request all the students, please utilize this opportunity, because maybe few colleges may conduct such type of webinars to the budding pharmacist. Because curriculum everyone will study. Those things will continue and will complete on time. But we should know, other than the curriculum, that is very important always. Okay, so please utilize this opportunity. Thank you, one and all.
Thank you so much, sir, for your wishes and blessings. Now it's time to invite Dr. C. Supriya ma'am to introduce today's speaker. Hearty welcome, ma'am. Please introduce the speaker to us.
Thank you, ma'am. Good morning to the respected correspondent sister, Reverend Sister G. Nirmala Jyoti Garu, Principal Sir, Dr. Pamal Reddy Sir, the faculty, students, and all the delegates who have joined in this international webinar. I'm glad to introduce today's guest.
Madam Supriya, madam.
Are in charcoal targeting
Your voice
insulin.
not audible, madam.
What, sir? It's not coming, sir.
Now, okay, madam. Continue.
Okay, sir. Okay. And sir has received Best Paper Presentation Award for his remarkable research publications. And sir also got Gates Scholarship during the year of 2007 to 9. Sir also worked as a member in project implementation group of DST FIST grant, which is worth of 60 lakhs. Sir also acted as editorial board member in ANU Pharmacy Journal and associate editor in Asian Journal of Pharmaceutical Research and Healthcare. Sir has guided around 30 PG students, UG students, around 8 UG students, and 1 PhD student as a direct supervisor. Sir has organized UGC, AICTE, and he is also organized various conferences and workshops as co-convener. He is the life member of Association of Pharmaceutical Teachers of India. Sir also recognized as nominee of CPCSEA, Government of India, New Delhi. So now I would like to invite today's guest speaker, Dr. Fani Kumar sir, to start the session on cellular and molecular mechanism of neuropeptides. And I'm very happy to introduce our guest speaker who has worked in our institution during the period of 2009 to 12. And before inviting the resource person, I would like to inform all the delegates who are attending either online or offline. Please, you raise the questions or you can post the questions through the chat box. And after the completion of this session, our guest speaker will I would like to invite today's guest speaker, Dr. Fani Kumar sir, to start the session on cellular and molecular mechanism of neuropeptides. Welcome, sir. Fani
Thank you, madam.
sir.
Thank you, madam. Thank you for your
Thank you,
nice
sir.
introduction. Am I audible?
Yes, sir.
Okay. Thank you, madam. So
Yeah.
first, I would like to thank all the members who are involved in this particular session organization, especially I would like to thank the Reverend Sister Dr. Shaurli Sister Garu and Reverend Sister Nimla Jyoti madam and the Principal Pamula sir and the Supriya madam and the organizing members Naresh sir and the Sri Kala madam. And good morning to all of you and welcome to today's webinar. And today's topic is on neuroscience, cellular and molecular mechanisms of neuropeptides. And today I would like to try to give some insights about the research work which we are doing at our lab in the University of North Dakota. So, yeah. So with this webinar, you are going to get some information about the following things like targeted brain regions. So I think in our brain, we have a different regions. I think everybody knows about anatomy and physiology. And at our lab and also in the USA, most of the people, they're trying to find out the different circuits which are involved in stress and memory and other behavioral activities. And the second part is the electrophysiology, the important technique which we are using to find out the electrical activity of the individual neurons to identify its function and how it is communicating with the other brain regions and also presynaptically and postsynaptically how they are communicating. And to identify this, we are using different resting membrane potential identification and action potential identification. And also we have idea about the inward direct. I think I guess most of the people have idea about the different potassium ion channels, leaking potassium ion channels and voltage gated potassium ion channels. Like in our body, in cardiac cells and kidney and blood and epithelial cells and brain cells, they're having some inwardly rectifying potassium ion channels. And in the next slide, I would like to give more information about the inwardly rectifying potassium ion channels and their involvement in the excitation of the neuronal cells. And finally, mechanism of action of individual neuropeptides. So this is what I'm going to give completely in this session. And for this, I have selected one hormone which we tried recently, that is melanocyte stimulating hormone. I think everyone knows about this melanocyte stimulating hormone. I think we read about this melanocyte stimulating hormone in endocrine system. And it is mainly from pituitary gland and it is also released in skin and it is also mainly located at the hypothalamus. And the melanocyte stimulating hormone function in hypothalamus was well established. So here, this melanocyte stimulating hormone, it can interact with the neurons located in the paraventricular nucleus. And with this communication, this melanocyte stimulating hormone can increase the glutamate release. And that glutamate release can stimulate the postsynaptic neurons. And they are involved in the control of appetite. This was well established. So after that, we are trying to find out melanocyte stimulating hormone, its influence on the MC4 receptor, and that is located in the dentate gyrus granule cells. I think everybody knows about this dentate gyrus, which is a gateway of memory. So this is located in the hippocampus. Okay, so this is the hippocampus structure. In this hippocampus, we have a important structure that is a dentate gyrus, and along with this dentate gyrus, this hippocampus is having coronary cell is a CA3, CA2, and CA1. So together, this is totally hippocampus. What is the importance of hippocampus? So this is the most important region for the memory. formation and also memory storage. So dentate gyrus is the initial part of this hippocampus, which is getting some information from entorhinal cortex. If you see this line, this is an entorhinal cortex, and this entorhinal cortex is having communication from the different brain regions, and it is going to send information to the dentate gyrus. And this dentate gyrus is divided into three parts. One is molecular layer, and the second one is the granular cell layer, and this granular cell layer is the important one, which is having the principal neuronal cells. They are called granule cells. And after that, this third layer is a hilus. It is also called as a polymorphic cell. And if you see this blue cell, the star shaped cell, this is the cell body of the granule cell, and these dendrites are located in the molecular layer. And from here, if you see, there is a fiber which is reaches to the CA2 region. I think it's impossible to find out on that large screen. So, but try to find locate this laser pointer. So from here, there is a fiber which is reaches to the CA3 and CA2 region. So initially from the entorhinal cortex, the information is processed to this particular granular cell, and from this granular cell, these fibers will carry information towards this corneaminous regions. Okay, so that's why this dentate gyrus is called as a gateway of information. So this is going to be an important part for the formation of the memory. And the CA3, CA2, CA3, CA4 regions, and these are important for the storage of memory. So means nothing but recall of memory, because already we have some stored memory in the CA3 regions. If you want to recall that, CA3 is involved. If you want to form a new memory, we need a dentate gyrus. So based on this hypothesis, we start to find out how this melanocyte stimulating hormone will interact with the MC4 receptor, which is located on the dentate gyrus granule cell. So for this, we have selected this particular article. This Du et al. They did some neuroanatomy with the help of immunohistochemistry. Okay, they have added some antibodies on the they have injected some antibodies into the mouse brain and also rat brain, and then they found that the dense MC4 receptor expression was they found the MC4 receptor expression on the dentate gyrus and also on CA3, CA4 and CA1 regions. So this is a beneficial area for the identification of this particular melanocyte stimulating hormone influence on MC4 receptor. Okay, the next, already this MC4 receptor activity was well known. So MC4 receptor and MSH complex will stimulate the G protein, and that G protein can stimulate the adenylyl cyclase, and this adenylyl cyclase could increases the cyclic AMP production. This cyclic AMP can increase EPAC and protein kinase A. EPAC is nothing but cyclic AMP activated protein molecule. So this PKA and EPAC, these two are also involved in the release of calcium, or maybe increase of some other cation levels to produce some contraction or relaxation in the normal things. But in the MSH and the MC4 in the neuronal cells, we are trying to find out after the cyclic AMP activation, how this EPAC and PK is responsible for the excitation of the postsynaptic neuron. Okay, maybe you will get a doubt. Already the mechanism was well established, but our title is representing cellular and molecular mechanisms of neuropeptides. So means still some other things, some other ion channels, they are involved in the active excitation of the neurons. Okay, so based on that hypothesis, we are trying to find out if MSH binds with the MC4 receptor, how this could impact the formation of memory and also the recall of memory. But here in this study, we are trying to find out only the new memory formation, nothing but the spatial memory identification or a normal memory processing with the help of dentate gyrus. And also we are trying in future this recall of memory by using this particular CA3 neuronal cells. So interestingly, this beta MSH along with memory activation, it could also have some impact on stress generation. So maybe there is a doubt like that, how this dentate gyrus could generate the memory and also could responsible for the stress. So here there is a difference, like a structural difference. the dorsal side of the dentate gyrus is responsible for the memory generation, and whereas the ventral side of the dentate gyrus is responsible for stress formation. So, but in this study, we are focusing on only dentate gyrus involvement in the memory. So, this could helpful for the development of new drug molecules, especially for the treatment of Alzheimer's disease or dementia-like diseases. And this is one aspect if you see as a pharmacy student. But if you see as a physiology student, so physiology is nothing but we are trying to find out the unknown things. So means melanocyte stimulating hormone is well known for the sleep. It is important for the sleep. It is also important for the skin pigmentation, and this is also important for the appetite regulation. But still, some other regions they are not identified what is the role of that particular melanocyte stimulating hormone at that particular location. So based on that hypothesis, our lab is focusing on to unravel the other the hormones which are already present in the brain, and there also reaches to the other regions, and where there how they're producing different responses with in interacting with the receptors. Okay, this is the main area of our lab. And then to find out this impact of melanocyte stimulating hormone and how it is increasing or how it is improving memory. So for that we have selected a procedure like a preparation of slice. It's like a normal process. We are trying to dissect the animal brain in a coronal section and the horizontal section to find out the individual cells. And the second one, the most important technique, electrophysiology recordings. So what we are trying to do, so here we are trying to um, dissect the brain, rat brain or the mice brain in a different variation, like a horizontal section or the coronal section. And after that, we are trying to identify the individual cell. I think this is an individual cell which is a viable or a live cell. This is possible under only higher microscope. And after the identification of this particular individual live cell, we are trying to patch this cell with an electrode, which is a glass electrode. It's nothing but our normal glass pipette. So we are trying to make it as a sharp with the help of the micro pipette manipulators by applying some heat, and that sharp edge we are trying to insert that electrode into the individual cell, and after that we are trying to apply some suction, and with that suction there is a formation of seal between the electrode and the cell membrane, and this could allow the movement of ions from inside the cell to this particular electrode, and this electrode is connected with the electromanipulators, and which is called as a patch clamp mechanism equipments, and these will convert this particular ionic movements into oscillations, and those oscillations will be converted into a signal, and that signal will be recorded in computer. So this is what we are doing. So this electrophysiology is most important for identification of individual cell responses with the help uh, individual cell responses towards the other drug molecules and also other peptides and also new drug molecules. Okay, so these are the two techniques we are using to find out the behavior of the individual hormones on individual cell. Okay, so this is the picture what we are doing in our lab. So this is giving information about the individual neuron. We are trying to patch the individual neuron with the electrode, and then we are trying to get information with the help of voltmeter. They had given, so this is giving only voltmeter, but we are using patch clamp electrode technique mechanism. Okay, so what we are doing means here the same thing which we have read in our first year anatomy and physiology in a nervous system action potential. So this is the pillar for this electrophysiology also, because the resting memory we are trying to find out the resting membrane potential of individual cell before applying the drug and after applying the drug. Okay, and how this could behave is does there any influence of the sodium ion or calcium ion or potassium ions? Okay, and also we are trying to identify the role of this particular potassium ions, especially inwardly rectifying. We are not trying to identify any voltage graded potassium ion channel activity. So we are trying to identify the influence of inwardly rectifying potassium ion channels in the excitation of individual neuronal cells. Okay, students. Are you able to focus? I don't know.
Sir, yes, sir. They are on the
Am
line.
I
sir.
audible? Okay.
Yes, sir.
Yeah. Yeah.
Yes.
Yep. So the next slide, which is giving total information about our lab, what we are doing every day. So we are trying to anesthetize the animal. Maybe we are using rat and mouse. And for this process, we are using ketamine and xylazine. After the anesthetization, we are trying to perfuse the brain, total animal body. So because this transcardial perfusion is very much important to remove the blood from the brain also. If the blood is present inside the brain, that will damage the individual cells, and we are unable to find out the live cells to patch and to produce electrophysiological recordings. And this is an isolated brain, and this brain is going to be dissected and it is converted into horizontal or coronal sections depending on your targeted brain regions. The first step which I had given, the targeted brain regions. Here our targeted brain region is a dentate gyrus, which is in hippocampus, which is easy to find out in horizontal section. So based on that, we are trying to dissect the brain in the form of horizontal section with the help of a vibrotome. And even in this vibrotome, we are using some external solution which mimics, or a cutting solution which mimics the this physiological salt solution like sodium, potassium, calcium, and magnesium, and all the oxygen and other things which are very much important to maintain the viability of the cells. After this, we are going to incubate the cells in a external solution under oxygen with a carbogen. So 95% is oxygen and the 5% is carbogen. So sorry, carbon dioxide, which is called as a carbogen. So after the 45 minutes of this incubation period, we are trying to focus that individual slice under microscope. And then we are trying to find out the individual neuron. And after identification of the individual viable cell, we are trying to trying to uh, trying to reach that cell with the help of a electrode, which is a glass electrode. So before reaching the cell, here we are trying to apply a mild positive pressure. We are injecting some air into that particular glass electrode. So that can push the cytoplasm into the cell. And after that, after reaching the cell, we are trying to pierce, or we are trying to insert this glass electrode into the neuron. And after inserting the neuron, after inserting this particular glass electrode, there could be a chance of formation of a dimple-like bubble. And whenever you see that dimple like bubble, you have to apply a brief and strong suction. Okay, that brief and strong section will form a particular complete patch between the cell membrane and the electrode. If you see here, the cell membrane, excuse me, so this was attaches to the individual regions, and here it will allow the movement of ions from the cytoplasm into this glass electrode. And here this glass electrode, which is attaches to the micro manipulator, and this could be easy to understand from this image. You can see here it is a neuron, and here this is a glass electrode, and this glass electrode attaches to the micro manipulator, and this micro manipulator is attaches to the amplifier, and this amplifier will convert these ionic signals into the oscilloscope, and these oscillations in the form of a wave, that waveform is recorded in this computer. And to better understand this total technique, I would like to compare this electrophysiology with our normal lab dose response curve. I think the fourth year B Pharm students and all the M Pharm and other students, they had an idea about this dose response curve. So for that also we are using a frog, and we are generally we are going for anesthetization, but we are not using any chloroform. We are using a breathing technique to anesthetize the animal to to produce numbness in the animal, and then we are going to isolate the rectus abdominis muscle, and we are going to keep that rectus abdominis muscle in a solution which contains a sodium, potassium, calcium, which mimics the physiological salt solution. Okay, so after that we are trying to add some drug molecules to find out the response in that particular skeletal muscle. So there is a chance. Before that, we are trying to take a baseline. I could explain that in the next slide. So this is what we are doing at our lab every day, and we are trying to patch the individual cell with a glass electrode, and then we are getting some data. from that particular cell before applying the drug and after applying the drug. Okay, so this is what we are doing in our lab. This is the procedure. Yeah. So coming to the results. So here we are trying to identify the beta MSH influence on resting cell membrane potential. So what is the resting cell membrane potential? Everybody knows that the resting cell membrane potential for individual cell is different. In cardiac cells, in ventricles is different, and the auricles is different, and also in sinoatrial node is different. Like in mostly in the neuronal system, the resting cell membrane potential would be at minus 70 millivolts potential, isn't it? Are you following, students?
Yes, sir.
Okay. So the resting cell membrane potential is minus 70 millivolts. So that would be due to these ions present inside the cell and outside the cell. So mostly at the outside the cell we have a sodium and the calcium ions and the chloride, and inside the cell we have potassium ions and other negative sulfur means proteins and others which are having a negative charge. So that is the reason we have the resting cell membrane potential at minus 70 millivolts. So after patching a cell, we are trying to take a control value. So this from this level to this level, which is a control value. So like how we are taking a response in control value in our dose response curve. I think if you did that one, I think it's easy to understand. Okay. So even in the dose response curve also, we are trying to get a baseline, and after the baseline, if it is stabilized, then you are going to add a drug molecule to find out its influence. Is it producing any contraction or not in skeletal muscle? Same here. After stabilizing the cell membrane potential, resting cell membrane potential somewhere at minus 70 or minus 69, it depends on the cell. So after stabilization, we are trying to add a beta MSH, and after addition of this beta MSH, we found that there is a change in resting cell membrane potential. So initially it would be at the minus 70 millivolts. If you see here, this is the total average of the 10 cells. So the control value would be at the minus 70 millivolts, and the beta MSH is all beta MSH resting membrane cell potential is at minus 60 millivolts. That indicates that almost there is a significant difference between the control and the beta message, and the beta message is having some influence in our excitation of the dentate gyrus granule cells. Okay, so this has given some insights about for the further studies, and after identifying this activity bit how this beta message is producing this response in the individual cell, then we are trying to find out the role of adenylyl cyclase in the beta MSH activity. Already we discussed about the mechanism of action of beta MSH, that is working with the help of cyclic AMP pathway. In the cyclic AMP pathway, adenylyl cyclase is the most important enzyme responsible for the formation of cyclic AMP from the adenosine triphosphate. Okay, so to find out that, we use the SQ22536. It's a chemical molecule which is blocking adenylyl cyclase. So we pre-treated the slices with SQ22536 chemical compound under carbozane, and after 30 or 40 minutes of the pre-incubation of the cells, we patched a cell, and after getting a stabilization, we had applied a beta message, and interestingly, we found that there was a decrease in resting cell membrane potential, which we observed previously. Without any chemicals, the beta message showed an increase in resting membrane potential. But in presence of the adenylyl cyclase inhibitor, beta MSH was unable to produce any response. So, and even these are the 10 to 15 cells of the SQ22536 treated, and it's almost like minus 66 to and maybe minus 65. But here, if you observe here, it is a minus 70 to minus 60. So almost the 10 millivolts difference is there. But here maybe a 2 or 3 millivolts difference is there. So there was a significant difference between the beta MSH alone and the adenylyl cyclase. So this indicates adenylyl cyclase is involved with beta MSH activity. Okay, we found that it is working. So the same mechanism is responsible for this particular activity. Still, I think you have a doubt. This is well known. Why again we are doing to find out this adenylyl cyclase activity? I would like to give that example explanation about that why we are doing, because we know that all the neuropeptides and the neurohormones and all the endocrine molecules which are present inside the body, they are well known and they are working with the help of different receptors and their mechanisms were already established. But our lab is focused on the one particular ion channel and how that could impact the generation of the resting membrane potential and also the excitation of the individual cells. Okay, and then we would like to find out this beta MSH is having resting membrane potential resting membrane potential change producing capacity, but we are interested to find out, does it have any influence on action potential generation or action potential number increasing capacity in individual cell? So for that we used an other protocol. That protocol is nothing but, so which is an excitation protocol. Means individually the each individual cell, they're not having any action potential producing capacity because their resting membrane potential is at -70, which is not suitable for generation of action potential. If any cell could produce an action potential only if it reaches to the threshold capacity, and after the threshold capacity if it reaches to the plus 30 millivolts potential. So that would be possible only whenever any molecule, biological molecule, if it binds with the receptor, if that could open sodium ion channel, then only it would be possible. But here we are trying to find out this action potential producing capacity by using a current injection protocol. So means we are injecting current with our software to that particular cell membrane. Okay, maybe you could get a doubt like how we are going to inject current means. So this total system is connected with the electrodes, and also there is a connection at the glass pipette also. So from there we are trying to inject some current to individual cell membrane. So here we are injecting the current in a increasing order, like a 40 picoamperes, 80 picoamperes, 120, 160, 200 picoamperes, like maybe 360, and individual cell is having a individual protocol number. Like if the cell is larger in size, we are injecting the current injecting picoamperes. If the cell size is very less, we are trying to decrease the current injection capacity. So based on that, we are trying to find out injected current, does it increase the action potential producing capacity or not. So from the left side, if you see here, this is the control, and here at the 20 picoamperes, I think there was no change in the cell membrane activity, and even at the 80, even at 120 and 140, almost up to 160 or maybe 140, there was no difference in the control, even we are injecting the current. So means to stimulate the individual cell at the resting cell membrane potential, so they need at least 160 picoamperes to produce action potential. Okay, and that action potential number is going to be increasing if you are increasing the current injection capacity. Okay, are you following, students? Because these are the most important techniques they are following to find out in almost all the labs, those which are involved with the physiology and also pathology and also some pharmacology. So electrophysiology techniques, I think it's the most important technique to get a chance in postdoc position or maybe in PhD position if you have a chance to explore this technique in India. Okay, next. So again, after administration of the beta message, so this is the control. I think there is a clear cut difference between these two. So after administration of the beta message, there was an increase in action potential, or there was a action potential generation within 40 picoamperes of stimulation, and you could differentiate the both of them at the 200 picoampere level. At the 200 picoampere, I think almost they are having a 10 around something, and beta message is double than the normal control value. So means that is indicating beta message along with the resting membrane potential changing capacity. It could also involve with enhancement of action potential number in individual cells also. So, and this is the diagram which is giving the comparison between the control value and the test beta message value, and there was a significant difference from 40 to 200, and we have injected up to 360 or somewhere around, but we didn't get any significance, but there was a significant difference between up to 200 picoampere value. So means they're having some influence on the action potential producing capacity also. Yep. So this is the main part in our lab. So they have influence in resting membrane potential. It is working with adenylyl cyclase, and it is also having influence on the action potential generation capacity. But the question is how this beta message is going to stimulate the dentate gyrus granule cell, because cyclic AMP that can increase the calcium level. The calcium is responsible, or any other ion is responsible for the excitation of this particular dentate gyrus granule cell. For that, we used an IV current. IV current is nothing but I is a current, V is voltage. Okay. So if you see this graph, the y-axis is a current, it's nothing but I, and the V is x-axis is voltage, it is a millivolts. So voltage is nothing but a potential energy. That potential energy which is important to promote the movement of current in and out of the cell. Okay, so there is a little bit confusion, but I would try to give my level best. Here it is a current injection, it's a picoampere. It's like activation of the injection of the current in the form of picoamperes. But here in this protocol, we are trying to inject an voltage current. Voltage in the potential means potential, and at that potential injection, the movement of the ions into and out of the cell we are going to record. So this black color, this is a control value. So after injection of the 40 millivolts, there was a change in some current, and at the 80 there was a change in some current, and at 120 there is a change. See, the current flow is started at the positive side, and after injection of the voltage potentials, the current entry is reaches to the negative side. So means each and every cell is having a normal changes. So means because we are injecting some potentials, because of that potential there is some change in current value. So here, sorry, black is a beta message. So here this is there is a change between the beta message and the control current process inside the cell. So this is clearly identified in this net current, which is nothing but subtraction between this control and the beta MSH values. We subtracted the values of both of them, and finally we got this image is nothing but a curve. So here if you observe at 0 level, there is a line which is intersecting this particular graph, which is indicating that at the 0 level, at the somewhere around the some potential level, there was no current inside or outside, means there is no movement of ions into or out of the cell. Did you get that point, students? This is most important to find out the potassium ion channels. So here we are trying to find out which ion is responsible for this excitation. For that we use this voltage and current protocols, and here we try to find out the movement of the ions into the cell in presence of the voltage. I would like to try to take a pen. Yeah. So for example, if this is a cell and here it is a cell ion channel or something like this. So if you inject a 40 millivolts potential, this 40 millivolts potential could increase entry of some ions into cell. So this ions is nothing but a ionic current is in the form of I, in the form of picoamperes. Okay, so here this is a potential which is in the form of millivolts. So if you are increasing this millivolts, there is an increase in ionic current entry into the cell. Did you get that point, students?
Yes,
I
sir.
don't know. Okay. Yeah. And then next, we are trying to find out the individual. Yeah. 1 second. Yep. Okay. So this ionic current entry in with the help of this millivolts potential, we identified with the help of, in presence of control, sorry, in control value and also in presence of the beta message. And here you can identify there was a difference between the control value and the beta message. So there is a change in ionic movement in presence of beta message. So after subtracting the values between these two, we got a net current value. So if you observe this net current is started from positive side and it is reaches to negative side. And yet one somewhere at minus 90 or somewhere, this is going to be 0. Means at this level, the net current is 0. There is no movement of ions into the cell or out of the cell. So means some ion which is responsible for this particular activity of the beta message is stopped, that was stopped because of beta message identification. So here this level of millivolts potential is at minus 90 millivolts. This is at minus 90 millivolts. I think it's easy to identify. If you draw a line from here, it's almost reaches this point. Okay, so the minus 90 millivolts is a reversal potential for the potassium ion. Is a reversal potential for potassium ion. What is reversal potential? The reversal potential is nothing but at some point the movement of the ions will reaches to opposite direction. At some point the movement of the ions will reaches to the opposite direction. What does it mean? So here we have a voltage gated potassium ion channels. So what is the role of voltage gated potassium channels? Does it reaches inside the cell or outside the cell? What is the mechanism of voltage gated potassium ion channels? Anyone? Does anyone knows about this one? What is the mechanism of voltage gated ion channels? Repolarization. When we are getting repolarization, if potassium is going out of the cell or potassium is reaches inside the cell.
Inside the cells.
Inside the cell. Where is the location of potassium? Potassium is present inside the cell or outside the cell? Generally, potassium is present inside the cell. So, sodium entry is a depolarization. Sodium entry is a depolarization. And after the depolarization, exit of potassium from the cell to outside, which is repolarization. Which is a repolarization. And this repolarization, if the potassium, if it reaches beyond the normal level, that is called hyperpolarization. That is called the hyperpolarization. But here I am telling, I am trying to give an information about the other potassium ion channel, which is not working with voltage. Which is not working with voltage. This ion channel is present on the cell membrane. Always these ion channels are responsible. What are those? The ion channels like here ion channels, they are responsible to maintain the resting cell membrane potential at minus 70 level. And these are very much important in the cardiac and especially in the brain, because if you, I think sometimes we are getting anxiety. This means every person will anxious for some situations, and during that time our heartbeat is going to be raised and immediately it is getting to normal level. And even if you are getting anxious and after that after some time you are going to get a relaxation like that. So all these type of changes, a sudden sabotagation or sudden changes in our mood or sudden changes in the heart rate are regulated with the help of the inwardly rectifying potassium channels. They are also located on the cell membrane, and always they are trying to control the resting cell membrane potential. And here the difference is these ion channels, the potassium ion channels, they are having only inward rectification, means they're trying to get inside. So voltage gated ion channels are trying to get outside with the help of voltage difference. And but this inwardly rectifying ion channels always trying to getting into the cell. So that is the difference between the voltage gated and the inwardly rectifying potassium ion channel. And this inwardly rectifying potassium ion channel is, that's reversal potential of the potassium ion channel is at the minus 90 millivolts. So with this experiment, we found that there is an involvement of the inwardly rectifying potassium ion channel with a beta MSH application. So beta MSH not only working with cyclic AMP mechanism, this is working with the other potassium ion channel inhibition capacity. See, if you see, for example, if the graph is started, this is inward, means the cell, the ion is trying to reach us into the cell, but at one point the ion trying to change its direction, it is getting out of the cell. Okay, so for example, if you take this direction is inside and this is an outside, so this would be possible only at the reversal potential of the individual potassium ion channel. This is a little bit difficult, but try to understand this one because this is most important. Okay, if you have any doubts in this, we can discuss. So we found that inwardly rectifying channel involvement of beta MSH, and then we try to find out which inwardly rectifying potassium channel is responsible for this, because in our body, in different cells, we have 7 different types of inwardly rectifying potassium ion channels. Does anyone know about the ATP sensitive potassium ion channels? Students, anyone know about the ATP sensitive potassium ion channels? Oh, okay. What is the mechanism of glibenclamide? Both your B Farm students or M Farm?
Students who are joined in the
Yeah,
online,
online.
or who joined through the online, you can give your answer through the chat box.
Okay. So here the top one, it is giving information about this image. This is ATP sensitive potassium ion channel. So insulin and our type 2 diabetic anti-diabetic drug molecules, all the oral anti-diabetics, they are working with the help of blocking ATP sensitive potassium ion channel, especially sulfonylureas, not all sulfonylureas drug molecules. So this ATP sensitive potassium ion channel, if you block this ion channel, there is an increase in action potential, means the resting cell membrane potential, it reaches to 0 millivolts. Okay, so initially it maybe it's at minus 70, and because of the blocking the ATP channel, it reaches to the positive side. So that indicates that inwardly rectifying potassium ion channels, always they're trying to send potassium into the cell, and they can maintain the membrane potential at minus 70. They're trying to block excitation. Okay, so those type of ion channels are different types: potassium transport channels, classical inwardly rectifying channels, G protein gated potassium channels, ATP sensitive potassium ion channels. Okay, here we are trying to find out among these 7 ion channels which channel is involved with this particular beta MSH activity. So for that we used a barium, because the divalent cations like magnesium, barium and polyamines in the central nervous system, I think the 4th years they know about that polyamines. So they are responsible for the regulation of these potassium ions. So if you apply a barium, so again here we applied the same protocol, resting membrane potential. After getting a control value, we applied a barium to the bath, and this barium chloride is it changed the membrane potential to the positive side like a beta MSH. So which indicates that some inwardly rectifying potassium ion channel is involved, because barium is a non-selective inwardly rectifying potassium channel inhibitor. It can block all these 7 types. It can block all 7 types of channels, like our drug molecules in the pharmacology, non-selective blockers and selective blockers. So this is a non-selective blocker. It can block all. After the application of barium, we are trying to apply the beta MSH in the same cell. But here beta MSH doesn't change much. This means there is no significant difference between the barium and beta MSH. That indicates that beta MSH producing excitation was already produced by barium. So means beta MSH activity is completely related to inwardly rectifying ion channel. It is not at all related to any other responsible markers like a cycle. Means they are responsible for that. But along with that, inwardly rectifying ion channel is most important to produce beta MSH activity. Okay, so we found that there was an involvement of beta MSH activity with a inwardly rectifying ion channel. But again, for specific to specify which ion channel is responsible, we try to find out the SEH23390, which is also a new chemical, which is a D1 receptor antagonist, and it is also a inwardly rectifying channel 3.1 inhibitor. Please follow this cursor, this inwardly rectifying 3.1 inhibitor. So even if you apply this SEH2390, there was an increase in resting membrane potential. So means that cell, each individual cells, they're having multiple ion channels or maybe some specific ion channels, but this granule cell, it has almost KIR 3.1 channel is also there. After the application of the SEH, we applied a beta MSH. Even after the application of this SEH, we found there was a significant difference between the SEH and the beta MSH. So that indicates that this KIR 3.1 is not involved with this particular activity. So means beta MSH activity is not involved with the KIR 3.1. Then which type of ion channel is involved with this one? Then we tried a KIR 7.1 inhibitor. Interestingly, here also beta MSH showed a significant difference in resting membrane potential. See, here we can observe clearly there is a change in resting membrane potential from this level to this level. So means that indicates that even this KIR 7.1 is also not involved in beta MSH activity. And then we found that the glibenclamide, which is an anti-diabetic drug molecule, which is a KIR 6.2 channel inhibitor. And this KIR 6.2 is also an inwardly rectifying potassium ion channel, which is present in dentate gyrus granule cells. Here, after the application of the glibenclamide, see the glibenclamide is there throughout the experiment. Means we are using a control value is a TTX, and here we are used a glibenclamide, which is a KIR2 channel inhibitor, and this KIR2 channel inhibitor is we applied continuously throughout the experiment. So here we found that after the application of the glibenclamide, after the saturation level of glibenclamide activity, we applied a beta MSH. We found that there was no change in resting membrane potential. Like a barium, here we observed that there was no significant change. So means barium is a non-selective and KIR 0.62 is a selective ion channel for the beta MSH activity. So beta MSH excitation in dentate gyrus granule cells is only with the help of the inwardly rectifying potassium ion channel 6.2. So here I think you will get a doubt. So if KIR 6.2 is responsible for the beta MSH activity, then what is the importance of cyclic AMP? And what is the importance of adenylate cyclase? Are they important or not? Students? Okay, post in the chat. Yeah. So we found that the inwardly rectified.
One second.
Yep. Yeah.
So we are requesting all the delegates to answer by unmuting your mic. You can give the answers to our resource person.
No problem, madam. Okay. Yeah. So KIR 0.62 is involved in this particular activity. So here the adenylate cyclase is important and cyclic adenosine monophosphate is also important, because without them this 3.62 inhibition is not possible. Okay, so then we are trying to find out, yeah, so the mechanism of action of beta MSH on the dentate gyrus granule cells. So here, the last one I am trying to give that information. So beta MSH and the melanocortin 4 receptor interaction, which can stimulate the GS, and that can stimulate the adenylyl cyclase, and then it can increase cyclic adenosine monophosphate formation, and finally it is increasing the exchange protein activated by the cyclic AMP, which is called EPAC, and the protein kinase A. And so here, this is the mechanism. Beta MSH is going to block the ATP sensitive potassium ion channel. It is trying to block the entry of the potassium. See, generally we have only voltage gated ion channels. They're having exit, means during repolarization only potassium is going out. But here this ion channel is different. Always it is trying to enter into the cell, and it is trying to balance the resting cell membrane potential at the -70 to calm down and to reduce the excitation in all the times. So excitation of long time or long duration is dangerous for that particular cell, and to control that we have a inwardly rectifying potassium ion channels. They are very much important in cardiac and the neuronal system. And now this is the research is going in throughout all the areas, even in kidney and also in blood cells and all the epithelial cells also. What is the influence of these inwardly rectifying potassium ion channels in individual cell regulation or excitation? Okay, so here still we have a doubt because we used an adenylyl cyclase inhibitor SQ, and that blocked the response of beta MSH. Is that good? Because adenylyl cyclase is important for beta MSH activity. And further, we have a doubt. Which one is responsible for this particular EPAC inhibition? So KIR inhibition. Does EPAC is responsible or PK is responsible? To further confirm this activity, we tried an HJC 0350, which is an EPAC inhibitor, which is an EPAC 2. Specifically, this is present in nervous system. EPAC 1 is also there, and EPAC 1 is in cardiac system, and EPAC 2 also shares both the locations. So for that, we selected an EPAC 2 inhibitor, HJC 0350, and after the so this HJC 0350 is a pre-treatment, means we pre-treated the slices with this one, and then we applied the beta MSH. And after the application of beta MSH, there was no change in resting membrane potential. Even there was a decrease in resting membrane potential. So that indicates that this inwardly rectifying ion channel KIR 6.2 is inhibited only with the help of, what is that? EPAC. So means EPAC is responsible for the inhibition of this ATP ion channel, and the protein kinase J is not having any influence on this particular ATP sensitive potassium ion channel. So this is the title justification: cellular and molecular mechanisms. At the cellular level, we have an idea about the receptor and the cyclic AMP, and if it reaches to the molecular level, which molecule is responsible for the inhibition of the ATP sensitive potassium ion channel? So finally, we found that EPAC is responsible. This is our investigation, and we found this one because our lab is fast to find out the beta MSH activity in the dentate gyrus granule cell and how it is producing excitation and which type of inwardly rectifying ion channel is involved, and that particular ion channel inhibition is related to cyclic AMP or adenylyl cyclase or EPAC, because this mechanism is a well-known mechanism for our pharmacy because we read everything about the cyclic AMP in general pharmacology. But along with that, still there are other molecular mechanisms to find out how finally they could reach us to the target and how they are responsible for the excitations in individual neuronal cells. So this is the mechanism of action of neuropeptide beta MSH in dentate gyrus granule cell. And this study would be beneficial to the other scientists, those who are interested to get or develop a new drug molecule based on beta MSH like structure, because today we are developing a peptides, and today we are using computational techniques to develop a new structures to find out the computer-aided drug ligand binding capacity, and then we are trying to identify their pharmacological activities. there could be a chance to use utilization of this particular topic, and maybe it is a new discovery. Maybe it could be possible to introduce in maybe in next generation textbooks to find out the inwardly rectifying ion channels and the role of beta mSH in memory, because we have a physiology of beta mSH like a skin pigmentation or appetite or something, and memory also they are represented, but how they are improving memory, there was no report still. So we found this one, and further we are trying to find out this in vitro activity, because even electrophysiology is an in vitro activity, but that activity, that electrophysiology technique is the most important technique to identify the activity of individual drug molecules. With that base data of the in vitro, we are trying to identify this beta mSH role in YMH, especially spatial learning memory, and also with the help of the water mesh, we are trying to find out. This is the stereotaxic operators. So we are trying to cannulate the targeted brain regions with the coordinates, and we are trying to inject the beta mSH in a micro or nanomole level. So it means a microliter level or nanoliter levels, and then we are trying to identify their behavioral influence with YMH and water mesh. So this is about the total presentation today. Yeah. Any questions? So please raise. If you have any questions, please.
Thank you, sir.
Yep.
Pani sir. Sir, thank you very much, sir, for accepting our invitation. And
no, thank you,
giving
madam,
the
but I don't know.
Sir.
I don't know how many people they get it, because
sir, actually on
it's like a
offline
drive.
around 100, on offline around 60 to 100 students are there, sir. On offline, and on online around 100 pharma faculty and students were joined, sir.
Okay.
Sir. Sir, once again, thank you, sir, for giving valuable information about the importance of neuropeptides on the metabolism, as well as clearly you said about the action potential and resting membrane potential, depolarization and repolarization. So that and all very useful for our pharma fraternity, sir, either faculty or the students, sir. So once again, thank you very much, sir, on behalf of our Nirmala College of Pharmacy Management for giving such an useful information. And before winding the session, I request all the delegates to raise the questions, to ask the questions to sir. I request all the delegates, if you have any queries, you can ask
feel free
sir.
to ask the questions. Otherwise, maybe you didn't get any information from me.
Sir, if
it's not related to any work. Yeah.
Sir, if EPAC inhibit all the potassium sensitized ATP, is there any side effects?
Yeah. So means that depends on side effect, is depends on the drug molecule, I think. So here we are trying to find out the mechanism of action. So maybe if you develop a new drug molecule which is working with that particular inhibition capacity, so there could be a chance to find out its influence, and maybe side effects would be possible to identify later on. It won't be possible immediately. Yeah, pharmacologically could be possible.
Okay, thank you.
Yeah, thank you.
Hello, excuse me, sir.
Yeah.
Yes, sir. The question is, other than the concept, there is, you are basically from US, right?
Basically from USA. No, I'm basically from India.
Okay. Sir, I want to go to abroad, sir, for PhDs. Is it possible to continue that?
Yeah, I would like to give that information based on the, I think, 1 second. Yeah. So I think these are the acknowledgements about my lab members. I'm sorry. Yeah. Those who are interested to get an abroad education, or those who are interested to do PhD or any other things, I found during my these 2 years of observation, I found some other techniques. They are very much important to get a chance in individual labs. Okay. So we are lacking behind these techniques because because of lacking facilities. Especially, we don't have any facilities in our South colleges. And even there are no facilities in university level also. But if you could find any possibility to get a hands-on experience of molecular biology techniques, they are very much useful to get a chance in PhD or to get a chance in any lab. And also those people who are interested in a software side, I think if you have an idea about the different softwares like R software, Python, MATLAB, and SAS, learning only these softwares wouldn't get any chances. I think if you apply these softwares in a bioinformatics or data pooling or data mining or big data analytics, if you have experience in these techniques, then only they will give us a chance. Okay, only PhD students, the professor will see your previous background or your previous research activity, and also they will try to find out how best you are going to match with their lab routine activities. Like if they are doing some other activity, some other research, and if your research is not possible to fit into that research, you may not get that one. So selection of that one is also very much important. And also, if anyone is interested to surgical techniques, so here I had given only MCAO, medial cerebral artery occlusion, because here in US the neurological disorders are common, especially the cerebral stroke is common. So MCAO model is a cerebral stroke model. So most of the labs in US, every medical university is doing this MCAO model activities. So like spinal cord, MCAO, or kidney surgeries, or whatever the surgical techniques, if you have some techniques in your hand, definitely you could get a chance. And also those who are doing PhD, at least they could have some publications. Okay, and this is most important. In every lab, apart from the normal activities, so PCR, RT-PCR, qPCR techniques they are using. If you have a chance, you could get an exposure to all these Western blot or qPCR, PCR, RT-PCR, and these softwares. If you learn about PCR, RT-PCR, you have a chance to get into a job in medical school and pharmacy, and also immunology, virology, and a lot of opportunities for these molecular biology techniques.
Okay.
Yes.
Thank you so much.
Yep. Welcome. Any other questions?
Once again, we would like to
Okay,
inform
madam.
all the delegates. If you have any queries, you can ask the sir. Sir is on the line. So this is the good opportunity to clarify your doubts. Sir has given clear information, either research-wise or academic-wise.
It's your choice, even for the academics also.
Yes, sir.
Even for our abroad studies also. Yeah, maybe that slide could be useful for you. Yeah. Okay, madam. Once again, thank you, madam, for giving me this opportunity. Sir. And please convey my regards to Shaurli sister Yaru.
Sure,
and
sir.
Jyoti sister. Yeah. And principal sir and all the faculty members, Manjula madam, Nari sir.
Yes, sir.
And Spandana madam.
Okay, sir.
Yeah. Okay, madam. Thank you. Thank you once again.
Sir, this is the time to felicitate our guest speaker, Pani sir. So in this regard, as sir is on the line, or as sir is far away from our country, we would like to give our appreciation certificate, and we would like to send our appreciation certificate as a token of gratitude on behalf of Nirmala College of Pharmacy Management. So, sir, soon we'll send our certificate, sir, appreciation certificate to you, sir. We'll post the certificate, sir. And once again, I request all the delegates to fill the feedback form just to receive the participation certificate. So we request we'll send the feedback link after the completion of this session. So please fill the feedback link to get the participation certificate.
Okay. Please share
Thank
that.
you.
feedback, madam. Thank you, madam.
Sure, sir. Thank you, sir.
Yep. Thank you all, my dear students.
Thank you, sir.
Thank you.
Thank you, sir.
Yep.
Hey.